Hello, everyone. Thank you for sticking around today. I appreciate it. This summer, I was able to work with Dr. Kramer and Dr. Sodinger, and we studied the interface between lion's mane extracts and metabolic regulation in a colon cancer cell line. So the first thing I want to talk about, what exactly is lion's mane? Lion's mane is a medicinal mushroom described for its unique health qualities and health and nootropic effects. It's commonly found in like these supplement stores where people who hear about these effects can go in and buy them hoping to get these results. And the unique qualities of lion's mane, it was traditionally used in Chinese cultures for digestive health and stomach disorders, but over time, as it's become more and more studied, it's shown to be involved in neuroprotection, cognitive enhancement, and anti-inflammatory and even antioxidant effects. So just a quick little background. I just want to go over what exactly lion's mane affects and the bioactive compounds of it. The primary compounds are haricinones and arenacenes, and that's a chemical structure, and that's just the mushroom of lion's mane. And the molecular pathways it affects is neurogenerative and cognitive enhancement through NGF expression, which is nerve growth factor. It also is shown to have anti-inflammatory effects as well through suppression of NF-kappa B and COX-2. NF-kappa B just promotes pro-inflammatory cytokines, and COX-2 supports the production of prostaglandins. So lion's mane suppresses these pathways, which makes it anti-inflammatory. However, the goal of this project was something completely different, and we wanted to look at different pathways involved in lion's mane. We wanted to see if lion's mane had any involvement in xenobiotic metabolism, cell cycle or apoptosis, and other metabolic regulators. And that's just the extract that we got of lion's mane. It was from Suzanne, which is locally found in Joplin as well. I just want to quickly go over the methods. We started with tissue culturing with ALICE-180 colon cancer cells. They were serum starved overnight, and they were treated with either lion's mane or vehicle. And the vehicle was dimethyl sulfoxide, and then the RNA was extracted. We then performed real-time quantitative PCR on the genes that we just discussed, such as drug metabolism, cell cycle apoptosis, and metabolic regulation. The last thing we did was we performed a reporter assay with similar receptors that Dr. Nguyen talked about to see which exact nuclear receptors was affected by lion's mane. Okay, the first thing we're going to talk about is lion's mane's interaction with the cell cycle. On the left side here is just a bunch of cell cycle regulators that we interrogated, such as TP53, which is known as the guardian of the genome, or cyclin D, which has shown progression through the G1 phase. And we found that TP53, cyclin D, c-Myc, and P21 all found like a modest 1.5 to 2-fold statistical significant difference in cell cycle, showing that lion's mane does have a statistically significant effect in terms of cellular stress and cell cycle regulation. However, that was not the story in terms of metabolic regulation. We combined it all together in terms of genes involving like mitochondrial biogenesis, ketogenesis, glucose and fatty acid metabolism, and even regulation of glycolysis. And we found that vehicle and lion's mane were very similar. We found no statistical significance. So I just want to show that lion's mane's impact is completely independent of metabolic regulation. I just want to show that as well. And it was a similar story in terms of xenobiotic metabolism and gluconeogenesis. The only gene involved in gluconeogenesis was PEP-CK, and there was no statistical significance there as well. But in terms of xenobiotic metabolism, we studied the P450 enzymes and genes involved in phase 2 metabolism. The P450 enzymes for phase 1 metabolism is just enzymes that add a hydroxyl group onto a lipophilic drug to make it more water soluble. And the phase 2 metabolism is just adding a conjugate to prepare for excretion in either the bile or the urine. And they also have a bunch of transporters as well that either the efflux of xenobiotics or in the bile. And through all of that, there was no statistical significance upregulation or repression in the genes involved in xenobiotics. So when clinicians prescribe these drugs, they do not have to worry about patients, the drug-herb interaction. Okay, the last thing we did was a reporter gene assay involving the specific nuclear receptors of PXR, LXR, FXR, GR, PPR gamma, PPR alpha with a common co-activator, PGC1 alpha. Just quickly going over the methods, these cells were transfected with these nuclear receptors. They were then starved overnight and then treated with lion's mane. The main thing I wanted to show out of all these nuclear receptors that we interrogated was the 10-fold induction in PPAR gamma. I just want to highlight that the reporter gene assay is very different than what actually happens in the human body. So Even with that though, the results of the tenfold induction of PPR gamma show that it may play a role in insulin sensitivity, adipogenesis, and even lipid metabolism. So just going over all the results that we covered so far, I just want to show that for lion's mane, there was no significant engagement in xenobiotic metabolism and the metabolic pathways at the doses that we tested in terms of lion's mane. However, in terms of cell cycle regulation, I would like to test a dose response in terms of see how high the induction can go even at higher doses of lion's mane to see if it is dose dependent. I just want to show that these findings were found in the LS180 colon cancer cells, and these findings may not generalize across all cell lines. We may not see these findings in common hepatocyte cell lines or even non-cancerous colonocyte cell lines. So the clinical significance, that's what's important. The use of lion's mane supports the safety and drug interaction, as clinicians do not have to worry about lion's mane affecting these drugs and their effects. They also may be complementary in metabolic issues, as they show no involvement in metabolic regulatory pathways as well. And some of the next steps for this project will be to perform RNA sequencing of the specific genes that lion's mane affects, and to see the exact mechanisms of the lion's mane's pathways. Now, I'd like to thank everyone on the slide with a special thank you to these 4 people, Dr. Kramer, Dr. Sodinger, Catherine Elliott, and Dan. They played a huge role this summer, and they play a huge role at KCU in terms of advancing research. I would just like to thank those 4 people as well. Thank you. And here are my references. Yes.
So with these being colon cells as well as with the cell cycle, I understand the targets there. But why were you investigating gluconeogenesis? Like what was the jump out reason to look
I
at
think
that?
it was just something that we wanted to interrogate just to see if it had any potential impact. There wasn't any like specific reason that we were involving. We were just trying to see any other pathways that lion's mane may affect. Yep.
I was curious, when you make these extracts, do you know the purity, whether or not for sure you've actually got your chemical of interest there, or, you know, it could be the active ingredient may be a minor element of those things that are actually extracted?
That's one of the difficulties of taking supplements to begin with, I think, is that with all these supplements that people may buy online or through these health shops, none of it is actually regulated. So you pretty much just have to believe the label with what they tell you. But some of them aren't even third-party tested, so they honestly could just be lying to you to begin with. So that's a difficulty with taking supplements anywhere, since they're not regulated.
There is a lot known about the active. So the answer is 4 steps in line, and yes, they are related, and they're relative.
I'm really curious too, and well done. This is a nice line of reporting, students piggybacking each other on this line of reasoning. You mentioned insulin sensitivity as a potential. Is there some literature that supports that? Are you going to follow up with any kind of glucose uptake, or what are we looking at?
I think that reported gene assay would need to be repeated. I did not see any literature in supporting the fact that lion's mane does affect this pathway. I would just like to repeat that reported gene assay to ensure that that's what's actually happening. And we don't know if that's going to happen in other cell lines as well.
That's why they call it research.
And there's some background that just that Cam didn't go into, that things we've worked on last summer in other experiments, that when you look at PPAR gamma and what the downstream metabolic pathways are associated with PPAR gamma, and some RNA sequencing we did in LS180s. It ties together, but it wasn't necessarily that we wanted to bring it all out for this talk either. So.
We got